Good evening. Thank you very much for the introduction. And I thought it would be a challenge for me, but after listening to Herbert, I think we mostly agree, so it will not be as much as a thing. I thought that Herbert would say that Parkinson's disease is a prion-like disease, and we should all be aware and be careful about this. This does not work. Okay. So this issue of prionization of neurodegenerative diseases, is it a fact or is it a myth? And I would strongly say that it is a myth. So the debate, which Herbert and I agreed, we shall limit it to Parkinson's disease because otherwise we spread too far away. So whether prion-like mechanism is responsible for Parkinson's disease spreading, this is the question. And after listening to Herbert, I could almost agree with whatever he said. And I hope that I had a second thought, and I thought, you know, what is it that we're talking about? And I would say that maybe he was right in saying that this is definitely correct, but this is not actually what he said. He was reluctant to defend this position that he was invited to defend. So he stepped back a little bit and said, well, let's see. What are the questions that I see facing us? So firstly, if we try to compare alpha-synuclein pathology to prion disorder, the question is, what are the alpha-synuclein species which are responsible for the toxicity and for the spreading? It's not just enough to say an alpha-synuclein. In prion disease, we know much better what is the species which is responsible. Then there is a question that I think we have heard already from Herbert, whether spreading implies also infectivity. It's not the same thing, so although many people think about it in the same way. And then are alpha-synuclein species specific from one synucleinopathy to another, or are there different synuclein species involved in MSA versus Parkinson versus DLB? And this is, nobody addressed this question, but I think it's a fundamental one. What is the mechanism of cell death in those alpha-synuclein spreading-based models? Can we extrapolate the results obtained in alpha-synuclein spreading-based animal models into human diseases? And I would say that the answer is no. And the other neurodegenerative diseases which are we will associate protein like Alzheimer and the others, Huntington's and so on, share the same spreading toxic properties of alpha-synuclein. Is it analogous? So the story is old, and already, almost 20 years ago that this question has been asked. Actually, Posner has suggested this even before he got the Nobel Prize, but he did not get the Nobel Prize for this. And maybe he thinks that maybe in the future he will, but so far I don't see much chance for that. So in prion, the prion disease hypothesis is that PRP is changed into PRP-SC, PRP-scrapy, and this PRP-scrapy in turn accelerates the conversion of PRP-C to PRP-scrapy and so on in a geometric line, so it will involve the whole brain. We know that PRP-scrapy is toxic and causes neurolysis. We know that neopil PRP-scrapy is toxic to neighboring cells, particularly neurons. So this we know for prion toxicity. We don't know that this is so, as I will show you immediately, for alpha-synuclein toxicity. Synucleinopathies, of course, we have different ones, so Parkinson's disease and DLBD, which may be different or maybe the same, but definitely multiple system atrophy is a separate one, where the infected cells are not neurons, or primarily not neurons, but rather oligodendrocytes. So there is a difference, and the explanation has not been provided of why would alpha-synuclein toxicity or spread behave in one way in this disease and in another way in a different disease. So the primary suspect in the prionization of Parkinson's disease is alpha-synuclein. So we think, or we are told, that there is misfolding of the normal alpha-synuclein, which of course occurs in every cell at a quite high frequency in the membrane, not buried inside somewhere, very superficial. And then the other assumption is that this misfolded alpha-synuclein will be released synaptically or at cell death, and will be taken up by the postsynaptic cells, and that the concentration of this alpha-synuclein species, which is a toxic, assumed toxic one, exceeds the potential of cells to eliminate alpha-synuclein. I think this is a critical issue, and I will stress it in a minute. So, unlike prion diseases, the toxicity of alpha-synuclein spread along certain pathways. Of course, this is not the case in prion diseases at all. They do not spread along certain pathways. There is selectivity of the cells sensitive to alpha-synuclein, to the misfolded alpha-synuclein. Neurons, but not glia, certain neurons, but not others. So there is this specific selectivity, which does not occur in prion diseases. And the conversion of alpha-synuclein from soluble monomers to aggregated insoluble forms is a key step in the neurodegenerative process of Parkinson, DLB, and MSA. So we know that there is local deposition of alpha-synuclein in certain locations in sporadic Parkinson disease, whether it's caused by one thing or another, dorsal motor nucleus of the vagus, olfactory bulb, etc., and consequently in other areas. So this is the spread. But the neurons involved are distinct chemically. So this is completely unlike prion diseases. Here they are either serotoninergic or noradrenergic or dopaminergic cells, which are sensitive to the disease and die in this disease. Although not all neurons die. So again, unlike prion disease, which the prions kill all cells completely eventually, in Parkinson disease, some cells die, but many others do not. So even one of the initial parts of the brain which are involved in Parkinson disease, as we've seen from Brack, the dorsal motor nucleus of the vagus, very few cells die there, even 20 years after the onset of the disease. This cannot happen in any prion disease. And the alpha-synuclein depositions, for example, which are Levy bodies or Levy neurites, are not necessarily toxic. In fact, many people think that the Levy bodies are a defense mechanism against the toxicity. So the fact that there are depositions there of alpha-synuclein does not mean that the synuclein is there. It does mean that there is a protective mechanism of the cell that will prevent the cells from dying in spite of having high concentration of alpha-synuclein there. Let me also say that Levy bodies do not contain only alpha-synuclein. They contain many other proteins that I will mention in a second. So what is not contested, I think, at least I will not contest it, is alpha-synuclein is present in various isoforms, monomers, dimers, oligomers, soluble and insoluble, fibrils, and so on. I'm not an expert in this, so I will accept it. The alpha-synuclein can be released from nerve endings, maybe. The alpha-synuclein can be taken up by some neighboring neurons. Has been shown in animals. I would not contest it. That alpha-synuclein can aggregate, probably true. That the mutated alpha-synuclein, for example, in those people carrying the mutation A53T in the alpha-synuclein gene, are more likely to aggregate and induce toxicity. all this is not contested by me. Also, that endogenous proteins are released, not only alpha-synuclein, not only in a disease state, that endogenous proteins are released from nerve endings and from dying cells, and these proteins can be taken up by postsynaptic cells. And this process has been known for many, many years. For example, the rabies virus spreads in this way. And horseradish peroxidase has been used by neuroanatomists for many, many years. And this is, again, exactly the same mechanism. So proteins can be secreted by some cells, can be taken up by the next cell. So this is a process that can occur. The question is that we have is whether this can be implied in the toxicity of alpha-synuclein. Also, I'm not contesting the assumption that endogenous proteins, not only alpha-synuclein, can be misfolded intracellularly. That's the fact that proteins are damaged through their lifestyle and are replaced by others. And so there is a machinery that will synthesize new proteins as the other ones are dying, are misfolding or dying in a way. But it must be so that the cell, all cells in the body, must have developed over evolution a process to eliminate these misfolded proteins. So there must be this process, and I don't think anybody could disagree that these processes occur and that there is a way that the cell has developed in order to eliminate or to control this thing. This can be ubiquitination or any other of several proteins. We still do not know which are the normal mechanisms that are directed at eliminating misfolded proteins, whether it's just the ubiquitin pathway or maybe several other ones. What is the capacity of this process? So is it a limited capacity? Is it a huge capacity? This is a critical issue. How strong is this process that will eliminate the misfolded proteins, alpha-synuclein or others? Are there specific mechanisms to eliminate misfolded alpha-synuclein? And are these mechanisms dysfunctional in neurons in Parkinson's disease? So nobody has suggested this. So this is an underlying assumption that there must be some dysfunction of these mechanisms, of these normal mechanisms that are needed in order to eliminate the misfolded proteins. And let me say again, to come to this, to this point that I made earlier, that in Parkinson's disease, it is not just misfolded protein which are deposited. There are many other proteins. So maybe the dying cell of whatever cause it dies, fails to eliminate the misfolded proteins, alpha-synuclein and others, and therefore they aggregate, and therefore they are deposited, and therefore we see the Levy body or Levy neurites. So this is in Parkinson. In prion diseases, cell-to-cell transmission does not place primarily through synaptic transmission at all. Completely different from the proposed mechanism in Parkinson's disease, but rather spread to neighboring cells irrespective of their chemical specificity. So a complete difference between Parkinson and prion diseases. And the issue of whether Parkinson's disease or other neurodegenerative diseases are in fact infectious has been asked before. So has been asked already in the late 60s by a guy called Carlton Gajuszek, who won the first Nobel Prize for the elucidation of the infectivity of Creutzfeldt-Jakob disease. And he showed that taking brain samples from people who died from Creutzfeldt-Jakob disease and injecting them into animals, particularly into apes and monkeys, produces the disease in these animals. And it was very much like the disease that occurs in humans. He also thought maybe this occurs in other neurodegenerative diseases, and he tried. And he tried it for ALS, and he tried it for Alzheimer's, and he tried for Parkinson's disease. Not once, not twice, several times over, and always failed to show infectivity. So here are some of the paper from the annals, from neurology, evidence against transmissibility of Alzheimer's disease in neurology, or ALS and Parkinson disease, and ALS PD complex of Guam, and so on. So this has been studied before in the best way that this thing can be examined. This is the excellent model because it is proven to be a good model because in Creutzfeldt-Jakob disease it occurs. And in Kuru it occurs. But it does not occur in the other neurodegenerative diseases. So this has been studied very much and not shown. And I think we've heard from Professor Budka about the results of this paper, which examined this place. So the question was whether patients, whether normal people who are injected with prion protein, with prion protein will develop Creutzfeldt-Jakob disease, and yes, they did. So these are the poor victims of the use of pituitary extracts in order to treat their growth hormone failure, and after injecting the growth hormone, they did grow very nice, but within a few years, many of them, maybe a couple hundred of them, developed Creutzfeldt-Jakob disease. This is because we assume, although it has never been proven, but it's accepted, that the pituitary from which this growth hormone was extracted contained or was derived also from patients, corpses of people who died of Creutzfeldt disease. It is much more likely that many of these people who contributed the pituitaries had Parkinson's disease. After all, Parkinson's disease is much more common than Creutzfeldt-Jakob disease. And yet, none of the recipients, none of them developed Parkinson's disease. After waiting for several decades, none of them developed it. None of those who developed Creutzfeldt-Jakob disease, of course, they died relatively early, but many others did not die. And none of them developed developed Parkinson's disease. So this is a very strong argument against the transmissibility of this disease. And that's what they say. In this unique in vivo model of human-to-human transmission, we found no evidence to support concerns that neurodegenerative diseases underlying hypotheses underlying AD and PD transmit disease in humans, despite evidence of their cell-to-cell transmission in model system of these disorders. And the story of the grafted neurons that Dr. Budka showed us, I think, for me, it's a mystery. I don't know why you bring it as a argument in favor. This is an argument against it. This is completely against it. I mean, these cells that were implanted, the implants into the basal ganglia, were denervated. They did not have any neural connections. So how did they get the alpha-synuclein from, I don't know, from substantia nigra? They could not have gotten it. So how did the presumed misfolded alpha-synuclein get into the grafted cells which are denervated? And note also that the changes that occurred in these neurons did not consist of what we typically see in patients with prion diseases, i.e. spongiosis. So here are the differences between the pathological features of spongiform encephalopathies like Creutzfeldt-Jakob disease and Parkinson's disease. Spongiosis is seen in spongiform encephalopathies. Why? Because of the spread of the prions from one cell to the neighborhood. This does not happen in Parkinson's disease. Astrogliosis occurs in spongiform encephalopathies as a reaction to this thing. Does not happen in Parkinson's disease. In prion diseases, there is no cell specificity. All the cells in the neighborhood will die soon, very soon. In Parkinson's disease, this does not happen. There is cell specificity. So there is cell specificity in Parkinson. There is none in spongiform encephalopathy. So the mechanism must be completely different. And of course, the clinical comparison is also shows marked differences. So the evolution is very rapid. Why would, if we talk about a prion mechanism for alpha-synuclein, why would it take 20 years or more for the disease from its beginning until the end? Why does it take only a few weeks or months at most in Creutzfeldt-Jakob disease until the whole brain is deranged, and here it takes so long? The phenotype is, of course, heterogeneous in spongiform encephalopathies because the disease is not specific, the pathology is not specific, but it's, of course, similar in patients with Parkinson's disease. That's why we call them Parkinson's disease. And, of course, the EEG is completely different. So this issue has been debated in CONI several years ago, and by Dr. Hilker and Jonathan Brocci and Yoav Chapman, and came up as a report in BMC Neurology several years ago. But I think that since then, in the past 4 or 5 years since this debate took place, we don't have any further evidence. So my conclusion is that many proteins are released from neurons in vivo and, of course, after their death, and this can be taken up by neighboring cells. All cells must have the capacity to detoxify misfolded proteins, and the deficit of this process has never been shown in any of the neurodegenerative diseases. However, there is a difference between such cellular processes and brain disease. This is not the same. The fact that there are these processes, okay, but this does not mean that they are responsible for brain disease. No evidence exists that Parkinson's disease is progressing prion-like and other diseases. And of course, we can talk about other diseases where there's also this spread, like maybe Guillain-Barré syndrome. So just because of the analogy, because there is this spread, should we also examine prion pathology in Guillain-Barré? I think this is also. So I think what happens is that, of course, there is this, so several important differences exist between Parkinson and prion diseases. The stepwise progression of Parkinson's disease can have many explanations. The prion theory attempts to explain the spread of the disease, not its origin. Not its origin. So once we have identified what is the origin, where the prion, where the synuclein deposits start, then we can ask why don't, or maybe the same process exists also in other neurons. Whatever starts in one place can start in other cells. We don't need the infectivity theory to explain it. So PD is not transmitted, is not a transmitted disease despite finding in experimental animals. So the experimental animals that we see are artifacts. These are usually transgenic animals in which you load the system with extreme amounts of alpha-synuclein. So you get an artifact there. This is a completely artifactual situation. This is completely unlike the human situation. It can very easily be hypothesized that by injecting so much alpha-synuclein, this will be in excess of the normal capacity of the cell to get rid of the alpha-synuclein. But in the normal situation, where it takes years and years for the development of disease, this should not be in excess of this process, or at least this has not been proven. So this is what we have to say, and my point is that prion-like mechanism is a relevant explanation for Parkinson's disease progression, still unproven. And I would even say, for me, unlikely. But the proposition that we have here is that for the disease in humans, it is still unproven. Thank you for your attention.
Thank you, Professor Korczyn.